Welcome everyone to week 12, lecture 11, where today we'll continue talking about neurobiology and genetics, but more specifically continue to talk about methodologies that allows us to study cells and mutations. Last week we focused on iPSCs, or induced pluripotent stem cells. This week we will introduce what is called CRISPR and also combine CRISPR with the iPSCs. So building upon the use of the iPSCs that we talked about last week, which of course were incredibly important in studying mutations in neurons in individuals with Alzheimer's, what we're going to do this week is consider genome editing, which is CRISPR, and how genome editing can be used to introduce targeted mutations into the human genome. This figure here provides a diagram of how we can use such technology. So we begin, of course, with the patient. We isolate somatic cells from that patient. This is the first step in the CRISPR process. By the use of specific transcription factors, we can then reprogram the cells from a somatic adult cell into what is called an iPSC. It is at this point where we can start to use genome editing to introduce deletions or exchange of specific bases. This way we can grow up neurons of specific types, for example, neuron, astrocyte, microglia, oligodendrocytes, and we can then study the introduction of phenotypes that are relevant, in this case, for example, to Alzheimer's disease. These cells can then be used for drug screening that we see here. So the generation of iPS cells, or induced pluripotent stem cells, is one way to surmount the problems of sourcing post-mortem brain tissue, which of course is an incredibly difficult organ to study in terms of its molecular and genetic behavior. As we heard last week, we are able to differentiate iPS cells in vitro into distinct cell types, including different types of neurons, and incomplete intergenetic epigenetic programming, however, can affect the results of these iPSC experiments. In addition, genetic diversity within and between populations, including the presence of common alleles, may alter gene expression and the pattern of many human genes. And I'd refer you back to EQTLs and their effect on enhancers and gene expression. So really, based on this, we ask the question, to what extent is the cellular phenotype of an iPSC influenced by the presence of protective and risk alleles in an individual's genome? Should certain risk alleles start to contribute to these very rare variants, and for example, we've looked at these in Alzheimer's, we can use CRISPR to start deleting or changing the sequence of these risk alleles to determine their interaction with these very rare but high penetrance mutations. I remind you of our lecture on melanoma, where we saw an interaction between the rare familial mutation in the gene CDKN2A with the more common polymorphisms in the gene MC1R. So CRISPR, it stands for clustered regularly interspaced short palindromic repeats, and we, or rather molecular biologists, have hijacked CRISPR, which is originally at home in bacteria, to create a toolkit for introducing specific sequence modifications into human cells. And it is this sequence-specific change of our human genome that is known as genome editing. So CRISPR-Cas9 in genome editing. The CRISPR-Cas9 system, and I'll come to Cas9 in a minute, what it does is it causes double-stranded breaks at specific DNA sequences. This system is bacterial in origin, and the use of our recombinant DNA technologies allows for the introduction of the Cas9 proteins, which are nucleases, as well as the CRISPR RNA, which targets the nuclease to specific sites in the genome. We are able to introduce this protein RNA complex into a mammalian cell. What this allows us to do is the site-specific cutting and modification of human genomic DNA. CRISPR is a defense mechanism in bacteria and archaea, and it confers acquired resistance to phage, which of course are an infectious particle for bacteria. It is defined by arrays of short direct repeats, and these are interspersed by what are called spacer sequences. In this diagram is summarized the CRISPR locus. So what we see first is here is an infectious phage particle at the cell surface. What happens is its DNA enters the cell, and in some cells, in some bacteria, the DNA of the invading phage, or part thereof, will be incorporated into the bacterial chromosome. It will be incorporated into what is known as the CRISPR locus, that consists of these other CRISPR-associated genes that we see here. This CRISPR locus will then be transcribed, shown here, and this RNA, which contains this, what's called a hairpin repeat that we see here, this RNA will be processed into short fragments. One of these fragments that we see here, coded in red, will be the same sequence as the invading phage and will target and destroy that invading phage. So in this way, CRISPR is a type of adaptive immunity for bacteria. Let's look in more detail at the CRISPR locus. First of all, the spacers. These spacers are generally unique. They have identity to phages and other extra chromosomal elements. As we saw in the previous diagram, it is fragments of DNA from invading organisms that are incorporated into the CRISPR locus in the form of these spacers. We see the Cas genes. These Cas genes encode the Cas proteins that have endonuclease and exonuclease activity, as well as other domains involved in transcriptional regulation. The leader sequence is thought to act as a promoter of the transcribed CRISPR array. So as I've said, the CRISPR-Cas locus functions as an adaptive immune system against invading foreign DNA. Let's run through that one more time. Here is our bacterial cell, and what we're looking at is just the CRISPR locus. We see an invading phage, and the DNA from this phage enters the bacterial cell. Most bacteria, of course, will die following infection by this particular phage. However, in rare cases, phage DNA can be incorporated into the CRISPR locus, resulting in a bacterial cell that is now able to fight off subsequent phage infections. What we see then is we see a spacer element derived from the invading phage incorporated into the CRISPR locus in the bacteria. This locus can be transcribed, and we now see that the spacer sequence is part of a mature processed sRNA transcript. It combines with the Cas proteins to bind and recognize the phage DNA or RNA and degrades the phage mRNA or DNA. So the integration of foreign DNA into the CRISPR locus involves Cas proteins, host polymerases, ligases, and recombination proteins. It is in effect a modification of the bacterial genome by the insertion of this invading foreign DNA. And in doing so, it creates a type of adaptive immunity for future infections of that particular bacterial cell. As I've said, the repeat spacer array is transcribed into a long RNA that we see here. Cas proteins recognize and process the RNA into small RNAs called sRNAs, and they contain a spacer and two half-repeat sequences shown here. These sRNAs are complexed with the Cas proteins, and they base pair in a sequence-specific manner with phage DNA, degrading the invading phage RNA or DNA. So in summary then, invading DNA, for example from a phage, is incorporated into the CRISPR locus spacer. This CRISPR locus is transcribed as a long pre-CR RNA, which stands for CRISPR RNA, that contains multiple repeats and spaces. Remember, it is these spaces that are unique and in this case is derived from a phage. The pre-cRNA is processed to cRNA that contains a single spacer sequence, one of which will be identical to invading phage, in other words, complementary to the foreign DNA. It combines with a second RNA species called TRAC-R-RNA, and this completes a targeting complex. In association then with the Cas proteins, this complex consisting of the CR or CRISPR RNA and the TRAC-R RNA combines to break and degrade foreign DNA.